An axial T1 weighted MR image of a normal wrist in a 30-year-old woman shows the flexor tendons, black circles, within the carpal tunnel. The median nerve, red arrow, appears as a gray triangular structure on the volar aspect of the tunnel. The volar retinaculum, white arrow, forms the floor of the tunnel and should be straight or concave. An axial proton density weighted MR image of the wrist in a 35-year-old woman with carpal tunnel syndrome demonstrates volar convex bowing of the volar retinaculum, white arrow, indicating crowding of the carpal tunnel. The median nerve, red arrow, contains a slightly elevated signal, indicating edema. The flexor tendons are not swollen and no masses are seen. These are magnetic resonance images, MRI, of a normal brain, left, and the brain of a person who has suffered a stroke. The arrow indicates the region of stroke damage. The plane of the image is indicated in the lower left of each film. Clear vision depends on how points of light, either emitted or reflected from objects, are refracted by the lens to form a clear image on the retina. Through the process of accommodation, the lens is able to change its shape slightly. Accommodation allows a normal eye to not only focus on objects that are near, but also on objects that are far away. However, the eye is often unable to focus properly. If the eye is elongated, light from objects far away is brought into focus in front of the retina, resulting in a blurry image. This condition is known as myopia, or nearsightedness. Myopic individuals often squint in order to bring faraway objects into better focus. Myopia can be corrected by a concave lens. Another common problem is hyperopia, or farsightedness, which is usually caused by an eye that is too short. In this case, light rays from closer objects are brought into focus behind the retina, resulting in a blurry image. A convex lens will correct this problem. A stroke may occur when there is an insufficient supply of blood to a brain region, or when damage to the vasculature in a specific brain region causes bleeding into the nervous tissue. In general, there are three types of strokes that occur. They are classified based on the type of vascular defect that causes them. Cerebral hemorrhaging occurs when a vessel bursts, causing bleeding in a region of the cerebral cortex. An embolic stroke occurs when small clots that formed on other parts of the circulatory system occlude cerebral arteries and cut off the blood supply to a part of the cerebral cortex. An ischemic stroke occurs when a narrowing or blockage of cerebral vasculature results in oxygen deprivation in a specific part of the cerebral cortex. The specific functional deficits observed in someone who has suffered a stroke can be correlated with the functions of the damaged cortical region. In a healthy brain, the nerve impulses from the cerebral cortex travel through the brain and cross over to the opposite contralateral side of the body. When a stroke occurs, damage to the cortex is usually unilateral, since information to and from the cortex crosses over the midline. The effects of a stroke on the left side of the brain occur on the right side of the body. A person who has suffered a stroke may experience paralysis that may involve the face, weakness or paralysis of limbs, often limited to one side of the body, loss of sensation that parallels paralysis, difficulty with speech, double vision, loss of vision, dizziness or vomiting, severe headache, loss of consciousness. The ability to sense rotational acceleration is achieved by monitoring sensory organs in the semicircular canals of the inner ear, called crista ampullarii. The hair cells in the crista ampullarii are key to the vestibular sense. Each hair cell has a large cilium referred to as the kinocilium, and many smaller cilia called stereocilia. When the cilia are bent toward the kinocilium, the firing rate of the hair cell increases. When the cilia are bent away from the kinocilium, the firing rate of the hair cell decreases. During motion, semicircular canal accelerates left. Cupula bends with the endolymph. Cilia bend away from the kinocilium. Hair cell firing rate decreases. During motion, semicircular canal accelerates right. Cupula bends with endolymph. Cilia bend toward kinocilium. Hair cell firing rate increases. During motion, semicircular canal accelerates left. Cupula bends with the endolymph. Cilia bend away from the kinocilium. Hair cell firing rate decreases. The sense of hearing results from conversion of mechanical energy in the form of a sound wave into electrical energy by the structures of the outer, middle, and inner ear. Sound waves from the environment impinge upon the ear canal where they are directed to the tympanic membrane or eardrum. The tympanic membrane vibrates at the same frequency as the sound wave. The vibration of the tympanic membrane is transmitted to the cochlea by middle ear bones called the malleus, incus, and stapes. These bones serve to reduce the amplitude of motion but increase the force exerted on the oval window. The sound wave is then efficiently transmitted into the paralymph of the cochlea. The hair cells contain many short hairs called stereocilia and one asymmetrically placed hair called a kinocilium. When the stereocilia are bent toward the kinocilium, the electrical rate of the hair cell increases. Bending away from the kinocilium decreases the firing rate. The electrical signal that results is transmitted to the temporal complex, resulting in the perception of sound. Three cranial nerves carry taste information from the tongue. The facial, glossopharyngeal, and vagus nerves each contribute sensory fibers to different regions of the tongue. You may examine which regions of the tongue are served by each of the nerves by clicking on a region of the tongue. The facial nerve carries taste information from the anterior portion of the tongue to the brain. The glossopharyngeal nerve carries taste information from the more posterior regions on the tongue to the brain. The vagus nerve carries taste information from the posterior tongue and oropharynx. Conscious motor activity is controlled by a specific region of the cerebral cortex called the motor cortex. Information coming from the periphery is processed by the sensory cortex and may be communicated to the motor cortex. The motor cortex may then process this information to direct the desired response. Within the brain, information is processed by massive neural networks. These functional groups process visual information, motor output, endocrine function, and other complex behavioral and homeostatic mechanisms. This animation shows how neurons within a network communicate, integrating inputs from neighboring cells and then sending that processed signal to other neurons. Output from a neural network may influence other parts of the brain and/or go directly to an effector organ such as a gland or muscle. The detection of specific airborne molecules, referred to as autogens, results in the sense of smell. As the autogen is inhaled, it moves through the nostril and into the nasal cavity. There it comes in contact with the olfactory epithelium. The olfactory bulb, which lies above the nasal cavity, receives sensory information from the olfactory epithelium. Within the upper nasal cavity, the autogens dissolve in the mucus lining, where they can interact with receptors on cells in a specialized region on the olfactory epithelium. In a reflex arc, sensory information is processed at the level of the spinal cord, resulting in action without involvement of the brain. For example, if one's fingers are burned by a candle, the hand that receives the painful stimulus is rapidly removed from the flame with a jerking motion. At the same time, the brain is receiving information indicating that a painful event occurred in the hand. It is important to understand that although the brain may perceive the painful stimulus, the reflex arc acts to retract the hand without direct input from the brain. The ability to sense head position is achieved by monitoring sensory organs in the saccule and utricle of the inner ear, called maculae. The hair cells in the macula are key to the vestibular sense. Each hair cell has a large cilium referred to as the kinocilium, and many smaller cilia called stereocilium. When the cilia is bent towards the kinocilium, the firing rate of the hair cell decreases. Within the macula, the cells are embedded in a gelatinous layer that is weighted by otoliths. Otoliths shift gelatinous material. Cilia are stretched. Dissimilar orientation of hair cells results in patterns of increased and decreased hair cell firing. The pattern of hair cell firing encodes head position. Otolith stable, basal hair cell firing. The detection of specific water-soluble molecules found in food or drink results in the sensation of taste. Specialized cells found in taste buds on the tongue contain receptors capable of interacting with molecules found in our food. When a taste receptor is stimulated, an electrical signal is produced by the sensory cell, resulting in an impulse which is transmitted to the brain and results in the perception of taste. To form images of our environment, light emitted by or reflected from objects must be focused on the retina. Some of the light reflected from the surface of a yield sign is directed toward the eye. Upon reaching the eye, the light is first refracted by the cornea to send it on a path toward the retina. To reach the retina, the light rays must pass through the lens where a small amount of refraction adjusts the angle of the light to form a focused image on the retina. Central to the sense of vision is the transduction of light energy into electrical energy by cells in the retina called rods and cones. Cylindrical-shaped cells called rods are responsible for the perception of light and dark. Conically shaped cells called cones are sensitive to red, green, or blue light. Four basic taste sensations are encoded by sensory receptors in the tongue. It is thought that different combinations of sweet, sour, bitter, and salty sensations are responsible for the perceptions of flavors that we experience. You may examine which regions of the tongue receive each sensation by rolling the cursor over the tongue. Astrocytes are star-shaped cells with many branching cytoplasmic processes, commonly found between neurons and blood vessels. They provide structural support and respond to injury of brain tissue by proliferating and forming scar tissue. This high magnification histological image includes several sensory neuronal cell bodies in the dorsal root ganglion. This histological image was taken from a sagittal plane through the eye. This is a histological image of the organ of Corti. The canals of the organ of Corti are found in a spiral arrangement, much like the shape of a snail shell. This histological image reveals a lamellated corpuscle of Pacini. Note the onion-like appearance. This is due to the presence of around 60 closely packed lamellae. This histological image was taken from a section that is perpendicular to the retina. It clearly shows how the retina is organized in layers. This histological image was taken from a transverse section of the spinal cord. It clearly shows gray matter, white matter, and surrounding spinal nerves. This is a high magnification histological image of 3 neurons located in the gray matter of the spinal cord. This is a histological image of a valate papilla. Notice the abundant taste buds lining the edge of the papilla.